Hi, I'm Jess.
And I'm Adriad.
And today we're presenting
Detecting the immune system response of a 500-year-old Inca mummy.
So this is how it all started.
Historical mysteries, such as the early death of King Tut, have been studied using forensic techniques such as DNA extraction and PCR. In this way, scientists were able to uncover why King Tut died at the early age of 18 and found out that it's most likely because of disease, such as malaria. Information like this is really important, and it could completely change the way we interpret historical events.
A mummy essentially is a human or an animal whose skin and organs have been preserved either intentionally or accidentally in various ways.
Previous DNA techniques could detect the pathogen, but that doesn't always mean that an active disease was present. Detecting the immune reaction, however, of the body to the pathogen does give us evidence that an active infection was going on.
Now, existing antibody-binding immunoassays need fresh samples, use small number of target antibodies, and give false negatives and positives. And nobody likes false positives.
So why is proteomics better in this application?
So proteins survive way longer than DNA and don't need PCR amplification, so that means less contamination. Also, we get more of a resolved picture of the immune response than immunoassays, as a broad spectrum of proteins is characterized from the small samples.
3 mummies were recovered from Lulailaco, a volcano in Salta, Argentina. 2 of them were young children around the age of 7 or 8, and one of them was a 15-year-old girl who they called the Maiden. These kids were sacrificed as tribute in a capocacha ceremony, so they were killed either by suffocation, hitting them over the head, or getting them drunk and burying them alive.
So the bodies were preserved really well. This was due to 3 things. There was the volcanic ash that inhibited decomposing microbes, the freezing temperature which inhibited decomposition, and the snow that kept it airtight. So there were various samples taken. There was a small blood-soaked piece of cloth that was taken from the boy. Mouth swabs were taken from both the boy and the maiden that contained blood and saliva. When it came to the young girl, she was struck by lightning after death, so they didn't use any samples from her. The samples that were collected, however, were stored separately and kept dry until the analysis to prevent oxidative and hydrolytic damage to the DNA.
In terms of proteomic analysis, the following was done. But basically what they did is carry out shotgun proteomics. The samples were sonicated and processed to purify them. Mass spectrometry with custom nanoliquid chromatography electrospray ionization source was used. Automated microcapillary liquid chromatography tandem mass spectrometry was used. MSMS was carried out on the 5 most intense ions from the original mass spec, and the spectra was searched against a human protein database. Confidence levels and false positives were also accounted for.
So, the boy's expression profile was used as a control to compare the maiden's profile to, and he was assumed to be healthy because he had no signs of respiratory disease, only trauma. Thank you. There was a Wilcoxon Mann-Whitney test that was used to compare the spectral counts, and the proteins were separated into inflammatory immune responders and all other proteins. To assume the disease in the maiden, she had to have elevated level of the immune system proteins, but there was no difference between the unrelated proteins for both the maiden and the boy.
DNA processing was also carried out. DNA extraction was done in a very sterile environment, and they did PCR with 4 sets of primers that targeted 16S rRNA, MTP40, and heat shock protein 65 of Mycobacterium species, Mycobacterium avium, and Mycobacterium tuberculosis. Negative controls were used, and none of them amplified, which meant that it was a good experiment, and gel electrophoresis was carried out. All the products were sequenced, and 2 positive buccal samples from the maiden showed a Mycobacterium species heat shock protein gene and a Bifidobacterium species heat shock protein gene. Bioinformatics was used to identify them with significant certainty, and they used statistical testing like BPP together with weighted Shimodaira-Hasegawa test to determine the chance of misidentification, which was low.
And this compared alternative phylogenies to the best phylogeny by maximum likelihood analysis.
The results for the boy overall seemed to prove that he was quite healthy.
He had no lesions, no mucus enlargement or respiratory infection, and he had a normal level of nasal mucus protein. All the presence of blood was pretty much due to trauma. What we did find was alpha-1 antitrypsin levels were high and the neutrophil defensin levels were low, and this meant that there was no lung infection. Significantly different results from the maiden in the inflammatory immune category. Now, the maiden, on the other hand, she had all kinds of problems.
It was found after analysis that her eyes and brains were quite intact and well preserved. However, she had a bunch of other problems. There was a radiolucent area in the upper lobe of her right lung, mucosal enlargement of her left maxillary sinus, which indicated sinusitis. She had a zoster-like lesion on her right calf and streaks of mucus under her nostrils. Mass spec revealed 3 times the amount of nasal mucus protein than the boy had.
So the proteins that were consistent with immune response to infectious disease were cathepsin G, neutrophil elastase, alpha-1 antitrypsin, and neutrophil defensin 1 and 3. These were because of things like inflammation and neutrophil infiltration of airways, degradation of virulence factors and killing bacteria, mycobacterial infection, and in-lung tissue inflammation.
Proteomic analysis found 2 groups of proteins which were consistent with severe lung inflammation. One of them was S100A9, apolipoprotein A1A2, and transthyretin, which are found in chronic and acute lung inflammation and used as monitoring biomarkers for pulmonary diseases. They also found vitamin D binding protein, serine protease inhibitor, which is serpin, and more TTR, which can be found in acute lung inflammation, specifically mycobacterial infections.
Now, presence of these proteins in the mouth swab provides strong evidence of response to severe respiratory bacterial infection caused by the mycobacterium. So in terms of the maiden, inflammatory and immune response proteins were high, where the spectral counts were 44 compared to the boy who was 0. Levels of all other proteins in the sample were quite similar. The disease etiology was determined by amplifying a heat shock protein gene using mycobacteria-specific primers and DNA from the mouth swab.
PCR and sequencing confirmed Mycobacterium species presence, as well as non-pathogenic Bifidobacteria species, and this was from the vomit prior to death. Phylogenetic analysis indicated a higher chance of the sequence corresponding to a pathogenic Mycobacterium avium bovis tuberculosis rather than a non-pathogenic Mycobacterium strain. Discussion! It was concluded that ancient Andean people suffered from mycobacterial infections.
Mycobacterium tuberculosis can survive in the body for long periods of time without actually causing infection. So detecting presence of the pathogen doesn't always mean active infection.
This study provides direct evidence of active antibacterial immune response at the time of death in the maiden, and these results were completely different in the boy who was healthy. Radiological examination of the maiden's lungs showed overinflation and trapped air, and these are common attributes of somebody who's infected with Mycobacterium avium or Mycobacterium tuberculosis. Potential forensic applications in the present time studies include characterizing the physiological state of the source of blood in criminal cases, and we can determine whether an individual is sick or is just carrying a latent pathogen.
Now, forensic proteomics offers a sensitive but less contamination-prone alternative to PCR for ancient or degraded samples. Unspecific or low-sensitivity immunoassays are no longer the only way to detect active immune response and infer infection in historical samples.
So Jess, what did you think about this paper?
Well, this paper publicizes an important new technique in applying proteomics in forensic studies. This will pave the way to many further discoveries in the near future and can even change some historical stories in some massive ways. What about you?
I think shotgun proteomics technique needs to be further tested in the immune response of older, more damaged samples rather than the fresh ones that they got that were well preserved. We need to really determine how effective this method is.
The technique that was used was great, but it needs to be taken one step further, which was done in a paper that was published earlier this year on January 4th. Usual forensics techniques at the crime scene include a mechanical or ballistic reconstruction of events.
Now they can take DNA from the bullet and see which subject was hit by which bullet. But it's hard to tell which bullet was the one that caused the lethal injury, and this is actually really important for legal proceedings. Now, with proteomics techniques, The samples can be taken from the bullets and traced back to the actual tissue and organ which they have passed through.
And that's about it.
Thanks for listening to our presentation.
What was I supposed to say?
We hope you enjoyed it.
Oh.